Hi everybody, welcome back. Today we're going to be talking about the plasma membrane and transport across the membrane. The plasma membrane acts as a barrier between the inside and the outside of the cell, and it's very important in controlling the flow of materials across itself. The nucleus is inside the cell and contains the genetic material of the cells, the chromatin. And between the nucleus and the plasma membrane are all of the organelles within the cytoplasm. Although this video does not cover the structure and function of organelles, you are expected to learn the structure and function of all the organelles. The plasma membrane is made primarily out of phospholipids. Remember from our macromolecule study that a phospholipid is a molecule that contains a phosphate head and two fatty acid tails. Taking a closer look at the structure of this molecule, you can see that the phosphate head has a negative charge, so it is polar. The tails are just long chains of carbons and hydrogens, so the tails are nonpolar. Our phospholipid molecule is therefore what we call amphipathic. One portion is hydrophilic and one portion is hydrophobic. This is important because the body has a very high water content. About 70% of the body is water. This means that the hydrophilic heads are happy because they are polar and they like interacting with water. They're hydrophilic. But the hydrophobic tails are not happy in water and they want to try and get away from the water in much the same way as oil and water will separate from each other spontaneously in your salad dressing. The phospholipids therefore form a lipid bilayer, which looks a little bit like a fat sandwich. The polar heads face the exterior and interior of the cell. The fatty acid tails are in the middle, making up the inner portion of the plasma membrane. This is a great solution because it takes very little energy to maintain the plasma membrane. The phospholipids are held together by hydrophobic and hydrophilic forces, and we don't have to waste energy forming bonds to hold these molecules together. The phospholipids form this bilayer spontaneously, and they are pretty free to move around within the membrane. Also present in the membrane are various glycolipids and glycoproteins. Glyco means sugar, so a glycolipid is a fat with a sugar group on it. A glycoprotein is a fat with a protein group on it. These molecules are used for signaling, transport, and identification in the cell. We have two main types of proteins that are associated with the plasma membrane: peripheral and integral. Peripheral proteins are attached to just the inside or just the outside of the membranes. You can see an example of a peripheral protein here. An example of a peripheral protein would be a cellular adhesion molecule. These allow transient binding of the cells to other things, which helps cells aggregate and form tissues. The other type of membrane protein is called an integral protein. This means that the protein intrudes into that inner lipid portion of the membrane. Some integral proteins go all the way through the membrane, and these are called transmembrane proteins. Others go just into the lipid portion. These are still integral proteins, but they're not transmembrane. These molecules are used for signaling and transport across the membrane. The layout of the plasma membrane means that some molecules can pass easily through the plasma membrane, while other molecules cannot. We call this characteristic semi-permeable. Small, nonpolar, hydrophobic molecules are quite comfortable passing through that hydrophobic core of the inner membrane. Examples of this are oxygen, carbon dioxide, and nitrogen. These are all nonpolar, uncharged, small molecules. Lipid-soluble molecules like steroid hormones and some vitamins can also pass quickly through the membrane. Other small molecules can pass through even though they are polar, like ethanol and glycerol. They're small enough to move through the membrane. However, large polar molecules are too big. Things like amino acids, glucose, and nucleotides. They cannot pass across the plasma membrane because they are too big, or they are charged, or they're too polar. Likewise, charged particles like ions cannot move through that lipid core. The ability of the plasma membrane to selectively determine what comes in and what doesn't allows it to form gradients. Let's talk about what a gradient is and why it's so important. Imagine I had a bottle of perfume and I sprayed it over on one side of the room. In the area where I am, there will be lots of perfume molecules. However, on the other side of the room, there is no perfume. You wouldn't be able to smell it because all of the molecules are on my side of the room. This is a concentration gradient, a change in the concentration of some molecules through space. After some time, you would expect that over on your side of the room, you might start to catch a whiff of the perfume. The molecules are moving around on my side, and eventually some of them will start to make their way over to your side. This is diffusion, or the movement of molecules from a greater to a lesser concentration. When molecules are moving from a greater to a lesser concentration, we say they are moving down the gradient or with the gradient. This is represented by the arrow. With no other intervention, the molecules will eventually even out, and there will be as many perfume molecules on your side of the room as on my side of the room. This is called equilibrium, and it means there is no concentration gradient anymore. It doesn't mean that there is no movement of molecules, it just means that there's no overall net direction. For every molecule that moves to the left, one will move to the right. We can classify types of membrane transport by whether we are moving them with their gradient or against their gradient. Passive transport is when you are moving molecules with their gradient from a high to a low concentration. Because some molecules can move right through the membrane and some can't, we have a couple of different types of passive transport, which we will go into in more depth in a moment. Active transport is the opposite. This is where we are moving molecules against their gradient. We're moving them from an area of low concentration back up towards the area of high concentration. This requires energy. Kind of like pumping water back up a hill would require energy. Let's talk more about the passive transport mechanisms. The first one is simple diffusion. In simple diffusion, molecules will move right through the plasma membrane down their concentration gradient. These molecules are generally small, uncharged, and nonpolar. These molecules will move down their gradient until they reach diffusional equilibrium, meaning the concentration is the same on both sides. For those molecules that are too big or are charged, they need some help to get across the membrane. They use a process called facilitated diffusion. These molecules still move down their concentration gradient, but they need some type of protein, either a channel or a carrier protein, to help them navigate through that nonpolar, hydrophobic environment of the inner membrane. A channel is like a tunnel from one side of the membrane to the other. Ions use channels to get across the membrane. Channels can open and close, meaning that sometimes those ions can get through the channel, but if it's closed, they cannot. In this way, the plasma membrane and the cell can determine who is allowed to pass across the membrane by opening and closing channels. It is worth noting that each channel is specific for a particular ion. The second type of facilitated diffusion is carrier-mediated. Large molecules like glucose will use a carrier to move across the membrane. The difference between a channel and a carrier is that a carrier protein will undergo a conformational change to bring the molecule across the membrane. The carrier, again, is specific for the molecule. For example, a glucose carrier will not be able to transport a different sugar across the membrane. The process of osmosis is specific to the movement of water. When the molecules cannot get through the plasma membrane, water will attempt to equalize the concentration. In the human body, most of the time the fluid in the body is a solution. In a solution, there is a mixture of two things, the solute and the solvent. The solute is the molecules that are to be dissolved. The solvent is the part that is doing the dissolving. In the body, the solvent is usually water. Let's imagine this drawing of a beaker represents a cell. You have the same amount of water on both sides of a semi-permeable membrane. However, the amount of solute, or stuff, is much greater on the right. Since the stuff cannot move across the membrane, the water will move in attempts to maintain the concentration of solute. Which way will the water move? You will see that the water will move to the right to dilute the stuff over there. When approaching this type of problem, you can remember that water always follows the stuff. Even though we haven't reached equilibrium, the water will not continue to move all the way to the right because of hydrostatic pressure. This is pressure exerted by a liquid on a membrane. This forces some liquid back to the left. In this manner, water moves to the right towards the stuff due to osmosis and is pushed back towards the left due to hydrostatic pressure. This continues until there is an equilibrium between these two forces. You might ask why we care about this, and it is because these forces can have disastrous consequences for our cells. When we talk about the amount of stuff on either side of the membrane, we have three ways to describe the concentration of solute. If the amount of solute on either side of the membrane is equal, the solution is called isotonic. If it is not equal, the side that has more stuff is called hypertonic. The side that has less stuff would be called hypotonic. In an isotonic solution, the cell's volume remains the same because although water is moving in and out of the cell, there's no net movement. However, in a hypotonic solution, there is less stuff outside the cell, so water will move towards the stuff in an attempt to dilute it. This causes an influx of water into the cell, which is where the stuff is. This can result in the cell swelling and perhaps bursting. When a red blood cell bursts, it is called hemolysis. However, in a hypertonic solution, there is more stuff outside the cell than inside the cell, and the water will leave the cell to follow the stuff. The cell will shrivel in a process called crenation, also not so great for the cell. Even small differences in tonicity between the inside and outside of the cell can have life-threatening consequences. This is the reason why we do not use water in an IV. We use a 0.9% saline solution, which is isotonic to the inside of the cell. The last type of passive transport I want to talk about today is filtration. This doesn't happen across the plasma membrane, but there are places in the body where we use filtration to separate particles based on size. This is very similar to using a colander to separate out items based on their size. When you use a colander, the small holes will prevent some things, like grapes, from passing through. But small objects, like couscous, can fit through the holes and will pass easily. Other materials, like rice, might not pass through easily, but increasing the pressure and essentially shoving those rice grains through the holes allows them to pass through the colander. In the body, we separate small proteins from larger proteins in cells. This is called filtration. The pressure pushing these objects through is the hydrostatic pressure of the blood. Increasing the blood pressure will increase the amount of filtration. Decreasing the blood pressure will decrease the amount of filtration. We'll be visiting this again later in the course. Everything we've talked about so far has been passive movement. It has all been movement down a concentration gradient, and it does not require an expenditure of ATP. However, in active transport, we are moving molecules the other way, against their concentration gradient. This means that they are moving from an area of low concentration to an area of high concentration, kind of like trying to pump water back up a hill. The most famous example of active transport is the sodium-potassium pump. This pump takes 3 atoms of sodium from inside the cell and uses the energy stored in the third phosphate bond in ATP to change the carrier protein and push sodium ions against their gradient outside of the cell. Then 2 atoms of potassium will bind to the pump and be brought inside the cell against its gradient. In this way, the cell can maintain a gradient for both sodium and potassium, high sodium outside the cell and high potassium inside the cell. This is called primary active transport because the hydrolysis of ATP is directly used to power the pump. Secondary active transport also moves molecules against the gradient, but in contrast to primary active transport, the energy is provided by harnessing a different gradient to power the pump. Remember that we established a nice sodium gradient on the last page? We can use that gradient to pull glucose into the cell. This carrier will allow sodium ions to flow down their gradient, but it requires sodium to bring along a molecule of glucose with it. Glucose is being pulled against its gradient. So this type of transport is active transport. We are not directly using ATP hydrolysis to power the movement of glucose. Because both molecules are moving in the same direction, these types of proteins are called symporters. In this next example, we are again using the power of sodium, but instead of moving another molecule in the same direction, we're moving calcium in the opposite direction. Sodium is moving into the cell down its gradient, but calcium is moving out of the cell against its gradient. It is active transport because we are moving calcium against the gradient. And instead of using ATP to directly power this pump, we are harnessing this nice gradient of sodium. Because the two molecules are moving in opposite directions, this type of protein is called an antiporter. Therefore, secondary active transport can either move molecules in the same direction with a symporter or in the opposite direction with an antiporter. It is active transport because one of the molecules is moving against its gradient. We can also move large particles into and out of the cell by one of three types of endocytosis. The first type is called receptor-mediated endocytosis. This is when a particle binds to a receptor and triggers the plasma membrane to invaginate and bring the particles into the cell. This is a specific interaction that requires a certain particle and a certain receptor on the cell. A more general form of moving large particles into the cell is called phagocytosis. The particle will be engulfed by the cell without a specific receptor needing to be triggered. Oftentimes, material brought into the cell by phagocytosis is destroyed by the fusion of the invaginated phagosome with a lysosome. The digestive enzymes within the lysosomes will lyse the particles. The last type of endocytosis is called pinocytosis, and this is when the cell is taking little teeny sips of the surrounding fluid. The vesicle again fuses with the lysosome, and the digestive enzymes within the lysosome will digest any solute. That's it for today. See you in class.